I want to tell you about a moment that changed how I think about one of the most common conversations I have with patients over 50. They come in for a checkup. Everything looks reasonable. And then, almost as a footnote, a little sheepishly, they say, "I still drink coffee. Is that okay? Should I cut back?" And for most of my career, I gave them the answer most physicians give, that it was probably fine in moderation, that the evidence was mixed, that if they were anxious about it, Decaf was a reasonable alternative, and that if they had heart palpitations or sleep problems, they should definitely cut back. I was not wrong about the palpitations, but I was comprehensively wrong about the overall picture. Because in 2017, a team of researchers published what is called an umbrella review in the British Medical Journal. Not a single study, but a systematic analysis of over 200 meta-analyses examining coffee and human health outcomes. They looked at everything. All-cause mortality, cardiovascular disease, cancer, diabetes, liver disease, neurological conditions, across dozens of countries and millions of subjects. The summary estimates indicated the largest relative risk reduction at intakes of 3 to 4 cups per day versus none, including all-cause mortality at relative risk 0.83, cardiovascular mortality at 0.81, and cardiovascular disease at 0.85. High versus low consumption was associated with an 18% lower risk of incident cancer, relative risk 0.82. Let me read those numbers again slowly, because they are extraordinary. 3 to 4 cups of coffee per day, compared to no coffee, was associated with a 17% lower risk of dying from any cause, a 19% lower risk of dying from cardiovascular disease, a 15% lower risk of developing cardiovascular disease, and an 18% lower risk of developing cancer, across more than 200 meta-analyses, across millions of subjects. And that is just the mortality data. Dr. William Li, whose research on the body's 5 health defense systems forms the foundation of everything we discuss on this channel, goes further. Coffee and green tea are what I call grand slammer foods. And what I mean by that is they activate all 5 health defense systems in the body. Not 2, not 3, all 5. Angiogenesis defense, stem cell regeneration, microbiome health, DNA protection, immunity. From a cup of coffee you are already drinking every morning. The question I asked those patients, "Is coffee okay?" was the wrong question entirely. The right question, based on the evidence, is how do you make sure you are getting the most out of it? That is what today's video is about. The exact compounds in coffee that produce these effects. The specific numbers from the strongest studies. Not the association data alone, but the mechanistic data that explains why those associations exist. The honest limits of the evidence. And the practical protocol that Dr. Li himself follows every morning. And why the order, the preparation, and what you add to it matters more than most people realize. Stay with me to the end. Because the section on decaffeinated coffee contains a specific preparation detail that most people have never been told. And the coffee plus green tea combination protocol in the final section produces mortality associations lower than either beverage alone. SECTIONON. What coffee actually is. The bioactive breakdown. Most people think of coffee as a caffeine delivery system. That framing misses 99% of what makes coffee medically interesting. A single cup of brewed coffee contains more than 1,000 different chemical compounds. The 5 categories that matter most for Dr. Li's defense system framework are as follows. Chlorogenic acids, CGAs, the primary polyphenols in coffee, comprising 6 to 10% of the dry weight of green coffee beans. Chlorogenic acids are the most abundant polyphenols in the standard Western diet for regular coffee drinkers, meaning coffee is, for many adults, the single largest source of dietary polyphenols they consume. Chlorogenic acids are potent antioxidants, reduce glucose absorption in the gut, improve insulin receptor sensitivity, and have direct DNA protective properties. They are also the compound that research at the University of South Carolina identified as having direct cancer suppressor gene activation. Chlorogenic and caffeic acid, bioactives found in coffee, changed cancer cells so that tumor suppressors in their DNA were unleashed, thwarting the ability for cancers to grow. Critically, chlorogenic acid concentration in coffee depends on roast level. Light roasts retain more chlorogenic acids than dark roasts. The roasting process degrades CGAs. If you are drinking coffee specifically for polyphenol content, lighter roasts are preferable. This is the opposite of what most people assume. Cafestol and kawal, diterpene compounds found in coffee oil. These compounds have demonstrated anti-angiogenic and anti-cancer activity in laboratory studies. They are found primarily in unfiltered coffee, espresso, French press, boiled coffee, Turkish coffee, because paper filters trap most of the coffee oil containing these diterpenes. However, caffeinated and cohort also raise LDL cholesterol in some populations through inhibition of bile acid synthesis. For adults with normal LDL cholesterol, the anti-cancer and anti-inflammatory benefits of these diterpenes from unfiltered coffee likely outweigh the modest cholesterol effect. For adults with elevated LDL or confirmed cardiovascular disease, filtered coffee, which removes most caffeinated and cohort, may be preferable. Caffeine, the most pharmacologically active and widely recognized compound in coffee, but not the primary driver of most of coffee's long-term health associations. Key point: many of coffee's benefits, including type 2 diabetes risk reduction and telomere length associations, are observed in both caffeinated and decaffeinated coffee, indicating that caffeine is not responsible for these effects. The polyphenols are doing the primary work. Trigonelline, an alkaloid that contributes to coffee's aroma and has demonstrated neuroprotective properties in laboratory studies, including potential protection against neuroinflammation associated with Alzheimer's disease. Melanoidins, brown pigment compounds formed during roasting that reach the large intestine largely intact, where they function as prebiotics. Research has shown that coffee melanoidins selectively feed beneficial gut bacteria, Bifidobacterium and Lactobacillus, while suppressing pathogenic species. The interaction of all 5 compound categories simultaneously, not any single one in isolation, is what earns coffee the Grand Slam designation in Dr. Lai's framework, SECTIONTWO, all-cause and cancer mortality. The full data analysis, the mortality data on coffee, is not from small or preliminary studies. It represents some of the largest and most consistent bodies of evidence in observational nutritional epidemiology. BMJ Umbrella Review, 2017. The benchmark analysis. At 3 to 4 cups per day versus no consumption. All-cause mortality relative risk 0.8395% CI, 0.83 to 0.88. Cardiovascular mortality relative risk 0.8195% CI, 0.72 to 0.90. Cardiovascular disease relative risk 0.8595% CI, 0.80 to 0.90. Cancer incidence high versus low. Relative risk 0.8295% CI, 0.74 to 0.89, representing an 18% reduction. Dose response note. In the most recent meta-analysis reviewed, 7 cups per day was associated with a 10% lower risk of all-cause mortality, relative risk 0.90, 95% CI, 0.85 to 0.96. But summary estimates indicated the largest reduction was associated with 3 cups per day relative risk, 0.83. The benefit plateaus and begins declining at very high intakes. 3 to 4 cups represents the optimal dose in the population data. Mediterranean cohort, long follow-up. Drinking more than 1 cup per day of caffeinated coffee was associated with all-cause mortality hazard ratio 0.5695% CI, 0.41 to 0.77, and cancer mortality hazard ratio 0.4195% CI, 0.20 to 0.86. At 18 years of follow-up, caffeinated coffee specifically was associated with all-cause mortality hazard ratio 0.5995% CI, 0.44 to 0.79. Decaffeinated coffee did not show the same long-term mortality associations. A cancer mortality hazard ratio of 0.41 means that regular caffeinated coffee drinkers in this Mediterranean population had approximately 59% lower cancer mortality compared to non-drinkers over 18 years. This is one of the strongest single cohort associations in coffee research. Japanese cohort. Cancer incidence. The adjusted hazard ratio for all sites cancer incidence was 0.7495% CI, 0.62 to 0.88 for coffee consumption of 5 or more cups per day in men P for trend less than 0.001 and 0.7695% CI, 0.58 to 1.02 in women. Coffee consumption frequency was inversely associated with mortality from all sites cancer. Large cancer mortality meta-analysis. About 4 million subjects. Investigations on potential modifiers for all-cause mortality in almost 4 million subjects revealed that the lowest relative risk for cancer mortality was present for those who drink 2 cups per day, RR 0.96, 95% CI, 0.94 to 0.99, P less than 0.0001. Colorectal cancer specifically. 2 umbrella reviews reported that consuming more than 1 cup of coffee daily may lower the risk of colorectal cancer by 11%. The association between coffee consumption and all-cause mortality in colorectal cancer patients was nonlinear, with an optimal intake of 3 to 5 cups per day. The honest scientific context. Essential for credibility. All of the above data is observational. Cohort studies and meta-analyses of cohort studies. Observational studies establish associations, not causation. Coffee drinkers may differ from non-drinkers in other lifestyle factors: diet quality, physical activity, socioeconomic status, despite statistical adjustments. No randomized controlled trial of long-term coffee consumption versus no consumption for cancer or mortality outcomes exists or is practically feasible. This limitation is real and must be stated directly. What strengthens the causal case despite this limitation? The mechanistic data is specific and well characterized. Chlorogenic acids activate tumor suppressor genes in cancer cells, documented at the molecular level. Cafestal and cahuiol have demonstrated anti-angiogenic activity in laboratory models. Coffee polyphenols reduce systemic inflammation and oxidative stress, measured in human blood samples. The biological mechanisms exist and are understood. The population associations are consistent across dozens of countries and multiple decades. The combination of biological plausibility and consistent large-scale associations is the strongest evidence structure available for long-term dietary exposures. SECTION3, liver cancer, the strongest single cancer evidence. The most consistent and robust human evidence for coffee's cancer protective effect comes from liver cancer research specifically. From a meta-analysis of 16 studies comprising 3,153 hepatocellular carcinoma cases, the summary RR for any coffee consumption versus no consumption was 0.6095% CI, 0.50 to 0.71. The risk of HCC was reduced by 40% for any coffee consumption versus no consumption. The summary RR for an increment of 1 cup of coffee per day was 0.8095% CI, 0.77 to 0.84. The dose-response relationship is one of the clearest in nutritional epidemiology. The pooled relative risks for liver cancer were 0.87 for 1 cup per day, 0.76 for 2 cups, 0.67 for 3 cups, 0.58 for 4 cups, 0.51 for 5 cups, and 0.44 for 6 cups per day. A linear dose-response relationship across the full range. For the most rigorous recent analysis, 18 cohort studies involving 2,272,642 participants and 2,905 cases plus 8 case control studies were included. An extra 2 cups per day of coffee was associated with a 35% reduction in the risk of hepatocellular carcinoma RR 0.65, 95% CI, 0.59 to 0.72. In a US-based pooling project of 1,212,893 individuals across 9 cohorts. Those who consumed at least 3 cups of coffee per day were at 27% lower risk of HCC-HR 0.73, 95% CI, 0.53 to 0.99, with evidence of an inverse dose-response relationship at a rate of 0.90 per additional cup per day. For any adult over 50 with metabolic syndrome, elevated triglycerides, fatty liver findings on imaging, or a history of alcohol use. This liver protective data is among the most practically relevant findings in this video. Coffee appears to protect the liver through multiple mechanisms, reducing liver inflammation, inhibiting hepatic stellate cell activation, reducing fat accumulation in hepatocytes, and promoting autophagy in liver cells. The protection is observed across diverse populations and appears to persist regardless of pre-existing liver disease status. SECTION4, type 2 diabetes, the dose response that removes caffeine as the cause. This section contains one of the most important mechanistic insights in coffee research. Multiple meta-analyses have established a consistent dose-response relationship between coffee consumption and reduced type 2 diabetes risk. Each additional cup of caffeinated coffee per day is associated with approximately a 6% reduction in type 2 diabetes risk. Each additional cup of decaffeinated coffee per day is associated with approximately a 6% reduction as well. The identical dose-response curves for caffeinated and decaffeinated coffee prove, not suggest, prove, that caffeine is not responsible for the diabetes risk reduction. The polyphenols, primarily chlorogenic acids, are the active compounds. They work by reducing intestinal glucose absorption, improving insulin receptor sensitivity, and modulating hepatic glucose production. This has two important implications. First, for adults who cannot tolerate caffeine for medical reasons, decaffeinated coffee provides equivalent diabetes protection. Second, it tells us that the polyphenol content of the coffee, not its caffeine level, is the primary determinant of its metabolic health benefits. This reinforces why light roasts, which retain more chlorogenic acids than dark roasts, are preferable for health purposes. SECTION5, DNA protection. The telomere numbers. This is the section that connects coffee most directly to Dr. Lai's DNA protection defense system, and where a large recent study produced findings that are genuinely extraordinary. Nurses' Health Study. 4,780 women. Telomere length. In this large study of coffee consumption and telomere length, we found significant linear associations with longer telomeres for higher total and caffeinated coffee consumption. We did not find significant linear associations for decaffeinated coffee consumption. Coffee consumption has been found to promote DNA integrity in intervention studies, including reducing spontaneous DNA strand breaks and protecting against chemical-induced DNA damage and oxidative DNA damage. 2025-2026 study. 5 additional years of biological age, drinking 3 to 4 cups of coffee per day, but not more, was associated with lengthened telomeres in study participants. Coffee appeared to be associated with approximately 5 extra years of biological age reversal in terms of telomere length measurements, 5 years younger at the cellular level, from 3 to 4 cups per day. The threshold matters. While coffee intake overall was associated with longer telomere length, increased intake of caffeine specifically was associated with reductions in telomere length. Individuals who exceeded the daily recommended intake of 4 cups per day showed a decrease in telomere length. The same dose-response relationship that applies to mortality. 3 to 4 cups optimal, diminishing returns, and potential harm above 5 or more applies to cellular aging markers. The mechanism: chlorogenic acids and trigonelline in coffee slow telomere shortening by reducing oxidative stress and inflammation. Chlorogenic acid inhibits protein glycation, the process by which sugars bind to proteins and damage them, producing advanced glycation end products that accelerate tissue aging. DNA strand break reduction from coffee consumption has been documented in human intervention studies with periods as short as 3 days and as long as 4 weeks, across consumption amounts of 600 ml to 1 liter per day. These are not correlational findings. They are direct measurements of DNA integrity before and after coffee consumption in controlled human studies. SECTIONSEX. The 4 defense systems in detail: angiogenesis defense, cafestol, and cawiol. The diterpene compounds in unfiltered coffee inhibit endothelial cell proliferation and migration, the processes that drive pathological blood vessel formation into tumors. Chlorogenic acid additionally suppresses VEGF expression in cancer cell models. The tumor suppressor gene activation from chlorogenic acids, documented at the University of South Carolina, directly prevents the transcription of genes that cancer cells need to recruit new blood vessels. Stem cell regeneration. The specific stem cell mechanism for coffee is less precisely characterized than for dark chocolate or omega-3 fatty acids. However, 3 indirect pathways are well documented. First, chlorogenic acids reduce systemic chronic inflammation. And as we have covered in previous videos, chronic inflammation is the primary environmental driver of stem cell depletion after 50. Lower inflammation means better stem cell preservation. Second, improved endothelial function from coffee polyphenols supports the vascular environment through which stem cells travel and operate. Third, the telomere lengthening effect from caffeinated coffee. 5 years of biological age reversal in the 2025 to 2026 study reflects improved cellular maintenance that encompasses the stem cell pool. Microbiome. Coffee melanoidins, brown pigment compounds from roasting, function as prebiotics in the large intestine, selectively feeding bifidobacterium and lactobacillus species. Studies have consistently found that regular coffee consumption is associated with increased gut microbial diversity. Chlorogenic acids modulate bacterial populations and influence short-chain fatty acid production. Coffee consumers show measurably different gut microbiome profiles than non-consumers, with associations toward the beneficial bacteria categories linked to lower inflammation and stronger immune function. Immunity. Chlorogenic acids reduce circulating levels of C-reactive protein, interleukin-6, and tumor necrosis factor alpha, the primary inflammatory cytokines that impair natural killer cell function and T-cell activity when chronically elevated. The reduction in chronic inflammation from regular coffee consumption creates a more favorable environment for immune cell function across all categories. Additionally, the metabolic improvements, reduced insulin resistance, improved glucose regulation, have indirect but meaningful immune consequences because insulin resistance is an established driver of immune dysfunction. SECTION7, the decaffeinated coffee question. Precise answer, doctor. Lie's specific position deserves careful analysis because it has important nuance. Dr. Lee advises against decaffeinated coffee because solvents are often used to decaffeinate coffee. The processing would also lower the beneficial compounds in the coffee. 2 to 3 cups per day of caffeinated coffee is health-promoting and recommended. The 3 primary decaffeination methods and their polyphenol impact. Solvent-based decaffeination, the most common commercial method, using methylene chloride or ethyl acetate. These solvents selectively extract caffeine, but also remove a meaningful portion of chlorogenic acids and other polyphenols. The resulting coffee has lower polyphenol content than caffeinated coffee. This is the method most commercial decaf brands use. Swiss water process uses water and activated carbon filters without chemical solvents, preserves significantly more polyphenols than solvent-based methods. Swiss water process decaf retains more of coffee's health-relevant compounds and represents the best available decaf option. Supercritical CO2 extraction, the most expensive method used primarily in premium decaf products, also preserves polyphenols well. Why caffeinated coffee showed telomere associations, but decaffeinated did not in the Nurse's Health study. Caffeine itself may have specific cellular mechanisms, including some effects on DNA repair enzymes and telomere-associated proteins that are distinct from polyphenol effects. The full explanation remains under investigation. The practical conclusion: if you can tolerate 2-3 cups of caffeinated coffee per day without sleep disruption or palpitations, that is the optimal choice. If you cannot, Swiss water processed decaf preserves more health benefits than standard commercial decaf. Standard commercial decaf, solvent processed, is the least desirable option in the category. SECTIONEIG8. What to put in it, and what never to add. No dairy milk. The same protein polyphenol binding mechanism that makes milk problematic in green tea applies to coffee. Dairy proteins bind to chlorogenic acids and significantly reduce their bioavailability and absorption. A 2025 study found that adding dairy milk to coffee reduced chlorogenic acid absorption by approximately 28% compared to black coffee. If you prefer whitened coffee, unsweetened oat, almond, or soy milk does not have the same protein polyphenol binding issue. No added sugar. Sugar in coffee directly contradicts the insulin sensitivity and glucose metabolism benefits that chlorogenic acids produce. The metabolic data showing 6% diabetes risk reduction per cup assumes black or near black coffee, not sweetened drinks. No syrup, flavored creamers, or sweetened milk alternatives. These products add 10 to 30 grams of sugar per serving to what would otherwise be a health-promoting beverage. A small amount of a high polyphenol spice is additive. Cinnamon, specifically Ceylon cinnamon, has demonstrated insulin-sensitizing and anti-inflammatory properties that complement chlorogenic acids. A small amount in coffee does not interfere with polyphenol absorption and may add a modest synergistic effect. This is consistent with Dr. Lie's food synergy framework, SECTION9. The Grand Slam morning protocol, based on the full evidence, here is the specific daily protocol the research supports: 2 to 3 cups of caffeinated coffee per day. This is the dose associated with the largest all-cause mortality reduction, RR0.83, the strongest telomere association, and the most consistent cancer risk reduction across multiple independent cohorts. 3 to 4 cups is the upper end of the optimal range. Above 5 cups per day, beneficial associations diminish and some markers, including telomere length, show reversal. Light to medium roast when possible. Lighter roasts preserve more chlorogenic acids than darker roasts. Dark roasts have more melanoidins but fewer CGAs. If you prefer dark roast for flavor, the melanoidin benefit partially compensates for the CGA reduction. Filtered or unfiltered based on cardiovascular status. Normal cholesterol. Unfiltered espresso. French press or pour over without paper filter preserves cafestol and caweol. Elevated LDL or confirmed cardiovascular disease. Paper filtered drip coffee removes most cafestol and caweol while preserving chlorogenic acids. The combination that produces the lowest mortality associations. The combination of less than 1 to 2 cups per day of coffee and 2 to 4 cups per day of tea was associated with all-cause mortality hazard ratio 0.7895% CI, 0.73 to 0.85. Cardiovascular mortality hazard ratio 0.7695% CI, 0.64 to 0.91. And respiratory disease mortality hazard ratio 0.6995% CI, 0.57 to 0.83. Coffee in the morning, green tea mid-morning. This is Dr. Lai's own routine. Espresso or coffee at breakfast, green tea later. The combination mortality data shows hazard ratios lower than either beverage alone because EGCG from green tea and chlorogenic acid from coffee work through different but complementary molecular pathways on overlapping defense system targets. 2 Grand Slam foods. 2 complementary polyphenol families. One morning. Let me close with the number I want you to carry. 3 to 4 cups of coffee per day. All-cause mortality relative risk, 0.83. A 17% lower risk of dying from any cause. 3 to 4 cups per day, associated with telomeres equivalent to approximately 5 years of biological aging reversal. Any coffee consumption versus no consumption, 40% lower risk of liver cancer. Each additional cup per day, 20% lower risk. And Dr. Lai's own words from his daily routine. Coffee is a grand slammer food. It activates all 5 health defense systems in the body. While you are hydrating, you might as well get a little extra out of it. You are already drinking it. You were already right. Tomorrow on Thrive After 50, we go deeper on walnuts. 2 servings per week associated with reduced colon cancer risk, slowed cellular aging, and a healthier microbiome. Plus, a specific finding about walnuts and brain structure that is one of the most striking nutrition-cognition connections in the scientific literature. Subscribe to Thrive After 50 so you do not miss it. I will see you tomorrow. All content is for educational purposes only and is inspired by the published research of Dr. William Li and the Angiogenesis Foundation. Thrive After 50 is an independent educational channel and is not affiliated with, endorsed by, or officially connected to Dr. William Li in any way. Always consult your own physician for personal medical advice.